Welcome, folks, to the second part of Chapter 11. So in our first part of Chapter 11, we discussed prokaryotes generally, and then we zoomed in on the domain Archaea and talked about some representative groups and species or genera within that category. And now we're going to begin our discussion of bacteria. So if we look back at this diagram of phylogenetic relationships, we can see, as we said, archaea are distinct in their phylogeny and they're a distinct domain, which we had discussed. And then within bacteria, we have a number of kind of groups and unique categories within. And so in this second part, we're going to talk about our deeply branching and phototrophic organisms here. And we're also going to talk about our gram-positive organisms as well. And then in the third part of the lecture, we're going to talk about gram-negatives. Okay. So deeply branching and phototrophic bacteria include cyanobacteria, but also some organisms as well, some other organisms as well. And then we'll talk about these gram-positives in a bit. So in terms of our deeply branching and phototrophic bacteria, these organisms give us an idea of how an early Earth looks like, because they have retained many of the characteristics that allowed them to kind of withstand and exist within an early Earth. These tend to be autotrophic organisms that live in unique environments. So for example, Aquifex is considered to represent some of the earliest bacteria. It's chemoautotrophic, hyperthermophilic, microaerophilic in its existence in the environment, where it derives energy and carbon from inorganic sources and lives in very hot habitats. So we're actually going to pause for a second and look at these three words: chemoautotrophic, hyperthermophilic, and microaerophilic. And actually what I want you to do is, as the primary discussion post for this chapter, I want you to dissect these words. This first one, you might look to chapter 6 for more information about. If you remember when we talked about carbon sources and electron sources, it'll give you some information to dissect this word here, and then hyperthermophilic and microaerophilic. And so you can put your word dissection there. That gives you some idea of what a prehistoric Earth looks like, although in your discussion post, you don't necessarily need to extrapolate that. That'll be pretty clear. Okay. Also, another really unique and interesting organism is Deinococcus. Deinococcus has the capacity to really withstand a significant amount of UV radiation. It's a chemoorganotroph, and it's similar in a lot of ways to gram-negative organisms, both in some of its anatomy, but also in its phylogeny. But it stains gram-positive because some of the unique characteristics that allow it to kind of be more resistant and resilient in its environment. It actually has pigments that allow for it to protect its lipids and its membranes, and also has a mechanism to actually repair shattered DNA from UV exposure. In fact, Deinococcus has been sent into outer space for 3 years, and then was still alive and able to replicate, reproduce, and repair its DNA upon re-arrival on Earth. So a really interesting organism there. Reminds me of this iron giant when it's able to kind of come back together on its own after it has been disassembled. And also this organism has been in outer space. So some very interesting, unique characteristics in that way. Okay. We also have our phototrophic bacteria that we'll talk about in this category. And these are organisms that are phototrophs. They don't have the complexity of eukaryotes that are photosynthetic, but they do have what we call photosynthetic lamellae, which are these surfaces or membranes within that allow for them to aggregate their photosynthetic pigments. You have blue-green bacteria previously known as blue-green algae, but algae is in reference to a protist, a eukaryotic protist, but these are clearly prokaryotes and are within the phylum Cyanobacteria. And these organisms have chlorophyll A as their pigments, whereas some of these other ones on here are green sulfur bacteria, green non-sulfur bacteria, purple sulfur bacteria, and purple non-sulfur bacteria instead have bacteriochlorophylls. But whatever the pigment, they're able to aggregate them and perform photosynthesis. Our blue-green bacteria are really interesting and unique, likely some of the primary contributors to the change over prehistoric time from a aerobic or low oxygen atmosphere to an oxygen that has a an atmosphere that has a significant amount of oxygen. Also likely the predecessors to eukaryotic chloroplasts. So here you're actually looking at plant cells with chloroplasts in them. When we look at the structures and genetic information within chloroplasts, and yes, they do have their own unique genetic information that is very similar to prokaryotes. They seem to have kind of a cyanobacteria type of origin. Also, these organisms have these colonies where they have some differentiation in their cell types. They're not multicellular organisms. These are just colonies of individual cells that are kind of still remain together in these filaments, most of them being photosynthetic, but some of them will actually do nitrogen fixation. And from a chemical perspective, the process of nitrogen fixation is actually a completely anoxic process, and it's inhibited by oxygen. And so to be able to distinguish and compartmentalize nitrogen fixation is important because you need to exclude oxygen from those cells. And so these cells are called heterocysts, and they're just a little bit different in their anatomy to allow them to do nitrogen fixation. You're also looking here at aconites. Aconites are a type of endospore that are produced by cyanobacteria as well. Okay. Now, our green sulfur, green non-sulfur, purple sulfur, and purple non-sulfur bacteria are a little bit unique. We already mentioned that they have unique pigments for photosynthesis. They also have completely anoxic photosynthetic processes, so they do not generate oxygen. And they often inhabit like anaerobic muds that are rich in different types of sulfur compounds like hydrogen sulfide. Our sulfur bacteria derive electrons from the oxidation of hydrogen sulfide and then produce sulfur. And then our non-sulfur bacteria instead derive electrons from the reduction of CO2 from organic compounds such as carbohydrates and organic acids. Our green sulfur bacteria, as we said, sulfur bacteria are going to produce sulfur. Within their photosynthetic processes, our green sulfur bacteria actually deposit that sulfur outside of their cells, whereas our purple sulfur bacteria deposit the sulfur inside of their cells. That can give them a purple hue, although not all purple sulfur bacteria are actually purple, just a note. So you can see them here. We're actually going to talk about these a little bit more near the end of the lecture and some associations that they have with sulfur worms. There's a really interesting kind of NPR piece about that interaction. So stay tuned for that. Speaking of which, I'm going to go back to our discussion of cyanobacteria really quick. I have linked in the description below a short documentary called Pond Life that predominantly talks about cyanobacteria, and you get to see some species of cyanobacteria. You get to see motile filaments. Actually, cyanobacteria will sometimes reproduce in motile filaments, which we had mentioned before, and just some really unique characteristics of them. So I definitely recommend you either pause the video now or at the end of this part, just look at that very short documentary. Okay. So now we're going to talk about gram-positive bacteria. And so they're kind of within gram-positive bacteria, there are two groups that kind of fall on phylogenetic or similar in their phylogeny. And if you think back to the structure of DNA, you have nucleotides GC and AT that tend to associate with one another. And so these two groups, one group tends to be low in its GC content in its DNA, and the other high in its GC content in its DNA. And then, so we'll talk about that in this part. And then just a note that when we get to gram negatives, GC content doesn't seem to be relevant in the phylogeny of gram negatives. So we won't kind of categorize organisms based on that in that discussion. Okay. So in our low GC gram-positive bacteria, we have three groups: Clostridia, Mycoplasma, and then this kind of aggregate of organisms, which is the other GC gram-positive bacilli and cocci. So in terms of Clostridia, these are organisms that are rod-shaped, obligate anaerobes, and a number of them form endospores. If you remember back to our discussion of endospores, it's a mechanism for self-preservation, and it becomes really important in a medical or an industry setting because it means that even after microbial control or sterilization, you might still have endospores present, and then those can then generate vegetative cells under the right conditions. And since some of these species actually have pathology, then you can actually have pathology and disease. And so organisms within the Clostridium genera specifically produce tetanus, or are the causative agents of tetanus, gangrene, botulism, and Clostridium difficile infections, which is like a severe diarrhea and tends to be an issue within hospital or nosocomial settings. I've actually, in the second link in the description below, I've included a video that gives us some information about Clostridium tetani, which is the causative agent of tetanus. I will warn you that it's somewhat graphic video, but it does give you an idea of how the organism is able to infect its host and kind of, how pathology occurs and gives you some idea about how these Clostridium are actually producing infection. Another interesting genus in this category is Epilopicium, which we had talked about before, one of the largest prokaryotic organisms that we know of that also has viviparity or live births. Okay. The second category is mycoplasmas. So myco is usually a prefix that we use for fungi. And that's not because it's a fungus, but because of its very unique pleomorphic shape under the microscope, where it has these kind of filaments or filamentous structures that kind of come out of the cell. And the reason for that is the second part of the name, plasma, because these organisms have actually lost their cell wall over time, over evolutionary history. They tend to be parasites, or they tend to live within a host, and so they don't need all of their protective outer structures. And so they just have a cell membrane instead of having a cell wall and a cell membrane. And so you can see that here, there are pleomorphic structures that give them their name. These are facultative or obligate anaerobes, and they're some of the smallest free-living prokaryotes that we know of, that they're about 0.2 to 0.8 micrometers in size. They tend to be, they tend to colonize mucosal membranes and urinary tracts of humans naturally and in a healthy microbiome, but they can opportunistically, or some species of which can cause pneumonia and urinary tract infections. Specifically, Mycoplasma pneumoniae produces what's called walking pneumonia, which is a milder form of pneumonia, or atypical pneumonia. And although it's hard to detect, it also has more minor symptoms than Streptococcus pneumoniae, which tends to produce more like a true pneumonia. Okay. As we said, it's lost some of its characteristic features because it tends to exist within a host. And so not only does it not have a cell wall, it also relies on its host for vitamins and nutrients and things like that. And so when we actually grow this in the lab, we have to supplement some of those vitamins and nutrients, even in a complex media, so that it will actually grow. When it grows, it has these unique fried eggs or sunny side up eggs colony morphology that you can see here, because the original colony forming units and its clones kind of almost grow into the agar, and then at one point they kind of just spread across the surface as well. Okay. This third category is our other low GC gram-positives. These are both bacilli and cocci, and so we'll talk about a couple different groups within. So genus Bacillus is important. It's common in the soil, and it can be, there's some species that can be pathogenic, but also some species that are beneficial within the environment. So anecdotally, you might know of Bacillus anthracis, which is the causative agent of 3 different types of anthrax: a cutaneous anthrax, which is most common and least lethal or fatal, inhalation anthrax, which is almost 100% fatal, and gastrointestinal anthrax of the stomach and the intestines. And that's actually the case study in your chapter deals with cutaneous anthrax. You also have organisms that are relevant to food production that you might know of just briefly through the discussion of genetically modified foods. So Bacillus thuringiensis is an organism that produces these crystalline structures that you can see in this image here, that are what are called a Bt toxin, Bacillus thuringiensis toxin, that essentially, as insects try to eat those bacteria, will actually is lethal to those insects, and then the bacteria can survive. And so what we've done is we've actually taken the gene for the Bt toxin and introduced it into most of our large-scale monocultured crops, even worldwide. And so this is one of the 2 most common genetic modifications of foods, is to have this bacterial gene for the Bt toxin incorporated into, for example, corn. And so what happens is when those insects try to, eat the corn plant or the corn fruit, then that crystal or the Bt toxin that's produced kills them and protects the plant. So of course, the discussion around GMOs is complex, but one thing that I just want to offer for you to optionally put in your discussion is, what is the second? I said there's Bt toxin is one of the 2 most common genetic modifications in our food crops. What is the second most common genetic modification in our food crops? Okay, so as an optional addition to your discussion post for this chapter, feel free to add that to D2L. Okay. Another thing about the Bacillus genus is that there are a number of species within this genus that produce antibiotics that we then utilize. And so remember that antibiotics oftentimes are produced naturally by organisms in the environment to allow them to outcompete other organisms in their communities. And so then we take them and mass produce them and then utilize them for our purposes as well. And so Bacillus species produce polymyxins and bacitracins as well. And we're going to talk more about antibiotics and the specifics and their modalities and so on and so forth, or their modes of impact in chapter 10. Okay. Also within this category is Listeria. Although Listeria does not produce endospores, it's a tricky organism in that it actually can still be active at low temperatures, and so it can be a contaminant of refrigerated foods, specifically dairies and meats. So anyone who during a pregnancy has been advised to avoid unpasteurized dairies or deli meats, this is why. Listeria rarely causes infection or severe disease in adults, but it can actually pass the placental barrier, and it can be fatal to the fetus. Also can be an issue for immunocompromised individuals, as it is a causative agent of both meningitis and bacteremia. Also in this category is lactobacillus. Most of our interactions with lactobacillus are positive. It's a non-spore forming rod that metabolizes lactose, and it's a normal part of the microbiome in the mouth, stomach, intestinal tract, and vagina. And it tends to protect those areas from opportunistic pathogens within the microbiome. Okay. And also it's important to food production. So pickles, yogurts, kimchi, things like that. Many of our different foods are produced in some ways by lactobacillus species. Also in this category are Streptococcus, Staphylococcus, and Enterococcus, all of which are able to produce enzymes and toxins that make them oftentimes problematic and pathogenic. Also, often, all of these are able to pick up genetic information from their environment, and that means that they can pick up antibiotic resistance. So all of these tend to produce nosocomial or hospital infections, secondary hospital infections, and be an issue because of their resistance to various or oftentimes multiple antibiotics. So you'll also notice from the name, Streptococcus, Staphylococcus. So Streptococcus has a cellular arrangement where you have that single file filamentous arrangement of cocci of spherical shaped cells, whereas Staphylococcus is just kind of a random cluster of spherical cells. Okay. So in terms of Streptococcus, like we said, it produces a number of enzymes and toxins that can lead to various illnesses. So typical pneumonia, Streptococcus pneumoniae is the causative agent of that. Also, pharyngitis or strep throat, scarlet fever, impetigo, various types of meningitis, wound infections, and other diseases. And as we said, many of them can be multi-drug resistant. Also, anyone who's been pregnant and has a GBS or Group B strep test at the end of their pregnancy, those are streptococci that actually exist naturally in the microbiome, but can infect a fetus upon birth and can be very pathogenic to a young child or a newborn. Also, Streptococcus mutans produces a sticky filament from sugars, and oftentimes is one of the predominant species within plaque on the teeth and within dental caries. In terms of Staphylococcus, again, as we mentioned before, it can be a normal inhabitant actually of the human microbiome, especially in the nasal cavity, but can be an issue, especially for cutaneous infections, and specifically can be multi-antibiotic resistant, like, for example, MRSA or methicillin-resistant Staphylococcus aureus. And in the lab, we will not interact directly with Staphylococcus aureus, but we will use some tests that allow us to understand more about the characteristics of Staphylococcus aureus or identify it in a medical or lab setting. Okay. And then you have these tables here, chapter 11, table 2, that gives you some overview of these organisms and some others within these categories. Okay, So that concludes our discussion of low GC gram-positives. Now I want to talk about high GC gram-positives, which exist within the phylum Actinobacteria. They are rod-shaped and also filamentous organisms, and they sometimes will resemble fungi and even produce spores, and so they're unique. And remember that spores or true spores are unique from endospores. So the three genera that we'll look at within this category are Corynebacterium, Mycobacterium, and Actinomycetes. Okay. So Corynebacterium are gram-positive, oftentimes pleomorphic, but sometimes rod-shaped organisms that produce through snapping division. And so if you remember, snapping division oftentimes will produce what V shapes or palisades under the microscope or that we see under the microscope. They are a normal part of the microflora in the mouth, skin, and urogenital regions, but there are some pathogens within this genus, specifically Corynebacterium diphtheriae, which is the causative agent of diphtheria. But we have few cases of this in the US because of the vaccine DTaP. So the D in DTaP is actually diphtheria. The second category is actually really unique. So Mycobacterium, named because of its mycolic acid in its cell walls. And so essentially these organisms have cell walls fortified with this wax called mycolic acid. And although it takes a long time for them to build up that wax, and therefore they are slow growers, that extra mycolic acid in the cell walls allows them to be protected against the immune system and even live within the cells of the immune system. And so these are intracellular pathogens oftentimes that live within the cells of our immune system or our white blood cells that actually try to phagocytose them, but then they are not able to because of their resistance to that. So very interesting. Also, because of that mycolic acid, we can't use a normal gram stain on them. Instead, we have to use an acid-fast stain, and you can see that here. That essentially allows us to stain that mycolic acid cell wall. And because they are intracellular pathogens, there are a couple of them that produce some unique pathology and are really global health challenges. So specifically Mycobacterium tuberculosis, which obviously causes tuberculosis, and then Mycobacterium leprae, which causes leprosy. And both of these are problems because essentially you can have latent infections where the host is infected with the bacteria, but it's actually residing within the cells of the host and not causing disease. And so it can show up later on. Okay. Then we have actinomycetes in this category. And so these are branching or filamentous bacteria, and they resemble fungi when you look at them under the microscope, but they are prokaryotes, not eukaryotes. Some important genera within them are Actinomyces, Nocardia, and Streptomyces. So Actinomyces are of the normal inhabitants of the mucosal membrane in mammals and humans, although there are a couple that are pathogenic. So specifically, Actinomyces israelii is a pathogen that destroys tissue, forms abscesses, and then it migrates to other vital organs within the body. Nocardia is important within soil and water, and it can degrade pollutants, which is great, but there are some species that do cause disease. And then Streptomyces are important both from an ecological and medical perspective in that they recycle soil nutrients and they degrade various organics within the soil. They're even able to degrade things like latex. And then they also produce some really essential antibiotics like erythromycin and tetracycline. Okay. And you can see them here, those filamentous structures and the true spores that they produce, as well as a reproductive mechanism. And so that concludes our discussion of gram-positive organisms. And so I would recommend that you look through some of the resources that are in the description below, and then come back to the third part of this lecture, where we'll talk about gram-positive organisms. I'm sorry, gram-negative organisms. So thanks for listening, and see you then.